Welcome, everybody. Today's lecture is Pests and Pathogens of Honeybees. Let's go ahead and get started. So, you've picked a very interesting time in US beekeeping to start your beekeeping career. And the reason that I say that is because in the early times of US beekeeping, mid 1800s through the mid 1900s, beekeeping was very different than it is now. Beekeepers during those times were primarily focused on the management of honeybees and actually managing the organism and not really dealing with a whole lot of major maladies of bees. So the idea is that you bought bees, you put them out to pasture, you super them up during the honey flow, you come and collect your honey at the end of the season, they overwinter through the year, and then you start back up next spring. So that was what beekeeping was back in I'll say the golden age of beekeeping in the United States. But you're in a very different era now. And this is the era of major global issues when it comes to pests and pathogens and the impacts that they're having on honeybees overall. So in the United States and globally, one of the major things that we're dealing with is varroa mite, and that is a global epidemic. And so a lot of what we do now in beekeeping is focused on the management of that organism and some other organisms as well. But varroa is by far the biggest thing we deal with. So a lot of what we do is really focused on the management of the pests and the pathogens that we're dealing with, and then beekeeping management as well. So now you have to be much more skilled than, say, your grandparents had to be when it came to beekeeping, because you're dealing with a whole set of issues and factors that they never dealt with back in those days. And so it's really important, as part of knowing that we are in that time here in US beekeeping, that you understand and know your pests and pathogens. And I want to implore upon you that this is not really, there's no beginner level for this. You need to take this very seriously as a beginner, and you need to study these things and understand them. And the reason is because this will impact your entire beekeeping career. This will make or break you. If you do not have a firm understanding of these pests and pathogens, you will not know how to react to them when you come across them, and you will absolutely. And in those situations when you don't know how to react or you don't know how to recognize something, the bees are who are going to suffer. So as a good beekeeper, as a good steward of bees, you should take this very seriously and feel it as a responsibility of yours to understand these pests and pathogens. And there's not a lot of them, so it's not a Herculean task, okay? There are very few, in fact. So you can do this very easily with just a little bit of studying, okay? Now, this is going to be an introductory lecture, but I want you to really understand that your knowledge should not stop here, because things are changing all the time in our industry. And we'll talk about one of those changes happening at the end of the pests sections of this lecture. But you need to know these organisms, and you're gonna be doing this. You're gonna be identifying them through hive inspections and visual ID most of the time. When it comes to pathogens, a lot of it is symptomology, so understanding the symptoms that you're looking at in order to understand what pathogen you're potentially working with, because you're not going to be able to visually see a spore of American foul brood. You're going to see the symptoms that American foul brood puts in a colony. And you also need to understand if it's serious enough for action. A lot of the things that we deal with, especially, and I'll use varroa mite as a prime example, we have thresholds that we have been established through research that tell you that if I have this many mites per 300 bees, I either don't have to do something or I need to do something when it comes to action. And we call that an action threshold. That's the threshold that we use to determine if action needs to be taken. You also need to understand your treatment options when it comes to these organisms that we're going to be talking about. And the main reason for that is that not all treatment options are relevant to you. Depending on where you live and the climate you live in, it's going to change which treatment options that are relevant to you and that you can use. And a prime example of that, I'll go back to varroa mite, is formic acid. So if you live in, say, Phoenix, Arizona, and you plan on using formic acid as a mite treatment during the summer, I can guarantee you're going to kill your bees, because formic acid cannot be used at high temperature thresholds. And so those are the things that you need to understand when it comes to treatment options. Now, for hive inspections, just to kind of, come back to what you should be looking at when you're doing hive inspections, you should be doing monthly inspections, at least every 4 weeks. 3 weeks is even better. You want to keep good inspection notes as well. It's important that you keep inspection notes because it gives you historical reference. You're not going to remember what happened 3 or 4 weeks ago. Make sure you're taking good notes because you can oftentimes really look at, you can create trends, over years and be able to determine when things are happening. A prime example, I'll use my own apiary. We've been doing this almost 20 years, and I can tell you almost within a couple weeks of when I am going to see my mite peak for the summer. And it's usually right at the beginning or middle of August, almost every single year. Some years the climate, like a couple years ago, we essentially had no snow up here all winter, and that changed things. But in a typical year, that's when I can expect to see my mite peak. So that allows me to actually prepare for that and do preventative actions where I never even have to use a chemical because I know it's going to be coming, and I know I can take some preventative actions to prevent that peak from occurring. I wouldn't know that if I didn't have my data points that I collected via my notes. When you're doing your inspections, you're going to look at brood condition, you're going to look at the pests, and you're going to note those in your inspection notes. You're going to look for pathogen symptoms and try to determine what you have, if you have something or if you don't. You're going to look at honey and pollen stores. You're also going to look at colony size and vigor. And that's a data point that's really important as well. That's where we count the number of frames that have bees on the front and backs of the frames. It gives us a relative size of the colony. That's important because some of the symptomology that has to do with some of these pathogens we're going to discuss, decline is one of the symptoms. And not all of these pathogens that we'll talk about will show themselves real evidently. And Nosema cerana is a prime example of a pathogen that can come in and really doesn't have a lot of symptomology with it, other than just a colony that's declining. So something to keep in mind is that vigor, that colony size, can actually inform what's going on with your honeybees if there's a problem. All right, so let's start with the pests of honeybees. And we're going to start with number one. The number one problem in U.S. beekeeping and beekeeping globally, for right now, because we've got another one on deck. But varroa mite in the United States is the number one problem. It is the number one thing you will be managing all season long. It should be on the forefront of your mind all season. This is not something we just deal with in the fall. This is something you manage all season long. The scientific name for this is very appropriate, Varroa destructor, and it is an ectoparasite, meaning that it is a parasite that is on the outside of the honeybee. Now, we know from research that the varroa mite feeds on the fat bodies of adult bees, and that has been the case. We've known that since 2019. And that was some research that Sam Ramsey did that really kind of blew the doors open on varroa and how it affects honeybees. So Sam identified that the bees in the adult stage, the mite is feeding on their fat body, which is actually located on the lower abdominal segments of the bee. And so that's oftentimes where the mite will reside on honeybees. Recent research in 2024 also, we learned that, in fact, that the mite is not just feeding on the fat body, which is what we had thought for several years after Sam's research, but in fact is also feeding on the hemolymph, but that's in the pupal stage. So during the adult stage, it's feeding on the fat body. During the pupal stage, which is the capped stage in the capped cell, that's when it's feeding on the hemolymph. And hemolymph is essentially bee blood. So not only is it feeding on the fat body, but it's also feeding on the hemolymph. So very devastating to honeybees because of the type of feeding that's occurring. Not only is it the feeding that has a devastating effect, but this opens entry points for viruses to come in. And that is another huge thing about varroa mite, is that it's a passive vector, well, for many viruses. Now, honeybees is, as far as Apis mellifera, the European honeybee that we work with, Apis mellifera never had varroa mite previous to about the 1940s. And that's why it's such a big issue for Apis mellifera, because it's not a co-evolved pest, meaning that there weren't eons of going back and forth in this kind of arms race between host and parasite, where they're constantly evolving these genetic mutations to outcompete one another. When you have a situation where something's co-evolved, and Apis cerana is where Varroa destructor came from, which is an Asian honeybee that's down in all sorts of parts of Southeast Asia and in Asia and larger parts of Asia, But during this time where Apis cerana and Varroa destructor were co-evolving, Apis mellifera was nowhere on the scene. It wasn't until the 1940s when we started importing European honeybees into Southeast Asia where we believe that this particular parasite jumped hosts. So in Apis cerana, because it's co-evolved, Varroa mite doesn't really cause an issue. Apis cerana has well evolved defensive mechanisms in order to handle Varroa. It can detect it in the hive. They will chew the Varroa mite up and spit it out, basically killing the mite. And they keep these Varroa mites at very low levels because they're co-evolved over a very long period of time. Because there's no co-evolution with Apis mellifera has, when this was first introduced to the European honeybee, Apis mellifera had no defenses to deal with this. It didn't even know that this thing was in the colony. And so it just runs rampant in Apis mellifera. And one of the biggest problems with this parasite is that it has a very fast life cycle. These mites reproduce every 10 days. Every 10 days, you are getting an exponential growth of mites in your colony. That is super, super dangerous, and one of the reasons why this is such a successful parasite. Let's take a look at the life cycle real quick. So we're going to start at the top here with this adult bee with Varroa on it. So this is the phoretic stage. This is the stage in which the adult mite is moving around. And this is a female mite that's mated. The male mites never make it out of the cell. It's just the females that have been mated that are on your adult bees. So we start with a phoretic mite here. What happens when the mite is on this bee, and this would be a nurse bee that we're talking about in this situation. Now, adults that come out from the landscape do bring mites in, but primarily you're going to find most of your mites on your nurse bees because mites reproduce in the brood. So they have to be near brood in order to reproduce. So the way that this happens is you have an adult bee that has a mite on it, and say a nurse bee that's working in your nursery. Right before the cap is put on the cell, so at the end of the larval stage, and as we start to go into the pupal stage and the pre-pupal stage, that mite will jump off of that adult bee and then bury itself down below the larva in that cell into the bee food. So it buries itself down there and kind of hides in there until the capping takes place. Once the cell is capped, then that mite starts to feed on the pupa. Now, the real interesting thing about this, like I said, this all happens really, really fast, is this female, this mite that went into this cell, she's going to start laying her first eggs 60 hours after that cell is capped. So this happens very rapidly. We're talking hours here. We're not talking days. And this is why when you have high Varroa mite levels, you have to act now, because this is every 10 days this cycle is taking place. So once we get these eggs laid in here, you're going to get about 1 to 6 eggs that are going to develop. And there's a couple immature stages you have here. The proto-nymph and the deuto-nymph are the two immature stages of the mite before it goes to adult. But what you have is in those first 5 to 6 days, you're going to have adult males that are going to be laid. And in the 7 through 8 days of that life cycle, you're going to have females laid. Once the females are laid, they start mating with their brothers. And so what happens is after the mating occurs within the cell, the gravid females, the ones that have been mated, are the ones who will come out with your adult bee when it hatches. And generally, it's one mated female that goes in and about 2 to 3 that come out. So you're doubling or tripling the amount of mites you have every time a female goes into a cell. This can get out of control really fast because of how fast this life cycle is. Now, a little bit of background on this. It was introduced in the United States in 1987, and there was a couple places where we think it came into, Wisconsin being one of them. This is something that you can see with the naked eye, but most of the time you cannot see it very well. It is about the size of a pinhead. It's orange and brown in color, and it's a little oval in shape. Now, there is a Varroa mite in this picture, and you might be able to see it because of how close you are to the bee. But the Varroa mite's located right there. It's under that abdominal segment that you see kind of tucked away. This is why we have to monitor for varroa mites. If someone's telling you, or if you've heard someone say, oh, I don't have varroa mites because I don't see them, you're never going to see them unless they're at explosive levels. And if you see mites crawling around a colony, it's already probably too gone to even recover from. So the reason that we do monitoring for this particular pest, meaning, and we'll talk about this, the alcohol wash or the powdered sugar roll, the reason we do that is because these mites tuck underneath these abdominal segments, and they're very hard to see. Even though you can see them with the naked eye, they're very hard to find in the colony. So we have to use a particular method in order to actually establish how many mites we have in a colony. That threshold that I was talking about, that action threshold, that's exactly what I'm talking about. So this is a really big problem. It's a big parasite. The bees it's very difficult for them to deal with it, and they are explosive as far as their reproduction rate. It's a recipe for disaster. To compound that, we know that varroa mites vector over 20 different viruses that heavily impact honeybees. Things like K-wing virus, deformed wing virus, a lot of different bee paralysis viruses. And these viruses affect the development of honeybees during their immature stage. So you have to realize that when these bees are impacted by this mite, oftentimes this results in deformed adults because it impacts them during their growth stage. And this is a prime example here. This is a drone that's suffering from deformed wing virus. And this is a very common virus. This virus is present in all your bee colonies. It just never has an ability to get into the bees until varroa creates a pathway by feeding on the outside of the bees, penetrating the cuticle of the honeybee. So with this particular virus, in deformed wing virus, this affects the development of wings. And what you see here are wings that basically look like they've been singed with a match. This bee will never be able to fly. This is a death sentence for a bee. And a death sentence for a superorganism if you have a colony full of bees that can't fly. That's a fundamental thing of honeybees, right? It's how they collect all their food. So really devastating these viruses can be to bees. Chemical camouflage is one of the explanations for why our honeybees have such a problem with varroa mite. So I mentioned the coevolution of this species with Apis cerana, and Apis cerana has developed a toolbox, essentially, of tools that it uses to deal with varroa mite because of coevolution. When organisms are not co-evolved, meaning they haven't had a long life history with each other, these adaptations take a really long time to sort out in the grand scheme of things. I mean, we're talking tens of thousands, maybe hundreds of thousands of years that these evolutionary processes take place. But what the problem with varroa mite and Apis mellifera, because it's not co-evolved, is that varroa mite has this really amazing ability that it has evolved because of coevolution with Apis cerana, where it actually has this property that we call chemical camouflage. What this property is that when a varroa mite comes into contact with the surface of a beehive or a bee, it takes on these compounds that we call cuticular hydrocarbons. These are basically smell molecules that are on the outsides of you, of me, of any organism. It's what gives us our smells, are these odor molecules on us. For varroa mite, when it comes in contact with a honeybee, it takes on the odors of that honeybee. And so essentially, European honeybees, the mite is invisible to them. They don't know it's even there, even though the mite is so big on that organism. This is a really savvy camouflage. I mean, it is literally you're chemically camouflaging yourself so that you're completely invisible to odor. And odor is one of the primary mechanisms that bees use to communicate and to understand their landscape. That's why the antennae are such an important organ in honeybees. Now, because we've had this mite in the United States since the mid-'80s, there has been a lot of concentrated breeding efforts. Now, this is one of the ways that we adapt bees to be able to deal with mites, is through selective breeding, which selective breeding is like evolution on steroids, essentially. It's where we are actually selecting the qualities that we want in these honeybees and then breeding them specifically for those qualities. And in nature, that takes tens of thousands of hundreds of thousands, maybe millions of years for that process to occur naturally. We can do that much quicker by getting involved and actually doing the selection. And so we have had many bees that have lines of bees that have come out in recent years that actually can deal relatively well with varroa mite. We have nothing that is a one and done, like a silver bullet, right? We have no silver bullets right now, but many of these lines of bees can deal with varroa in a much better way than bees that are not have not been selected for these qualities. So what I mean is, like, Italians, at large, Italian honeybees have not been selected for varroa sensitive hygienic traits, which is what we call these things when we're talking about these adaptations or these evolutionary traits that these bees have taken on over time. So Purdue ankle biters would be a prime example, a line of bees that was bred by Purdue specifically to deal with varroa mites. Varroa-sensitive hygienic bees are widely available in all sorts of different lines. Russians are another line that are known to deal with varroa mites very well. And just to throw this out there, Africanized bees, for those of you that live in the Southwest, this is something we dealt with when I lived in Southern California. These are very aggressive bees, and people do get killed by them. Animals every year. When I lived in Southern California, someone's dog got killed by Africanized bees. Although they do get a bad rap for the aggressive behavior that they have, many people keep Africanized bees. I've worked with beekeepers in Costa Rica. This is what they keep, is Africanized bees, because that's what they have. And so Africanized bees, if you are in an area of the United States that has Africanized bees actually deal with varroa very well. So something to consider. May not be the most fun bee to keep, but that's something to think about. Is, there are lines of bees, and I'm not suggesting that everybody go and buy Africanized bees. That's not what I'm saying at all. It's definitely, if you've ever kept them, it is not a fun experience. You get beat up a lot. But it's an example of, other lines that have these adaptations to deal with mites a little bit better than others. So this is why queen selection is so important when it comes to, after you've gotten started and kind of got your feet wet, making sure that you're setting yourself up with success with good queens is part of this whole equation. Now, how do we monitor for mites? We do that through our monthly checks, our regular inspections, and we do a check every single time we do a hive inspection. And the way that we do this is with the sugar roll or the alcohol wash. Now, there are other methods that have come out in recent years, the CO2 method. Ether method used to be the old method. The most reliable of all methods that we have are the sugar roll and the alcohol wash, and the most reliable out of those two by far is the alcohol wash. So if you want an accurate sample, do the alcohol wash. It is also the most foolproof of the two methods. But essentially the way it works is both methods are going to use about 300 bees, which is a half a cup of bees that you'll collect. And then you're going to use either the powdered sugar roll or the alcohol wash to remove the mites from the bees and then count the mites per that sample. And what this gives you is an ability to actually create a sweet little mathematical equation to determine what your percent infectivity is. So if you have 9 mites per 300 bees, that's a 3% infectivity level. 3% infectivity is the highest threshold that we have. So if you're mid-season, mid-honey season, and you're seeing 3% infectivity, this is when a treatment is absolutely needed. If you're on the back end of the season or the front end of the season, that number should be even lower. And we'll talk about that in another lecture. Now, the way that we deal with mites is we don't just use chemicals to deal with mites. I am not a proponent of using miticides unless you absolutely have to. And that's part of being a good beekeeper. We do this through integrated pest management, and this is the integrated pest management triangle. We're going to go through this in detail in the Managing Varroa Effectively lecture. But essentially what I want you to get from this is that we have different levels of control. So the ways that we're trying to manage these mites, they start at the base of the triangle and move up. So when we're dealing with varroa mite, we're always thinking about prevention. The best solution for varroa mite is to make sure you're taking preventative measures to set yourself up for success. So if we look at the cultural mechanisms, that's things like resistant stock. So those varroa sensitive hygienic bees like Purdue ankle biters, Minnesota hygienics, pole lines, Russians, these types of honeybees, they have been bred specifically for mite resistance, meaning that they can handle mites in a pretty good fashion. It doesn't mean that you're not going to have to do treatments. That's not what I'm saying, because we're still trying to get bred bees that can 100% deal with mites. We don't have that right now. But using resistant stock gives you a leg up. For those of you that are plant people, it's the same as buying like a plant that's resistant to like powdery mildew or something like that. It's been bred to be resistant to a pathogen. By purchasing it, it's going to have a less susceptibility to it. It doesn't mean that it won't be susceptible. Same thing with varroa mites in our resistant stock. Brood breaks are also an example of a cultural mechanism that we use to hamper mite development. We'll talk about that in detail in the Managing Varroa Effectively lecture. Mechanical means, that's the next level of prevention that we use. That's the everything below that red line is prevention. That'd be things like mite trapping, so removing drone brood frames, things like that. And then if those don't work, you've put them into place, and you get to the end of the season and you eventually hit that threshold, that's when we implement chemical measures. So you don't put chemicals in a colony unless you have a justification for it, which is your mite count. I get calls all season long, every single year, from beekeepers who are telling me that I don't have varroa mites, but I don't know why my bees look like crap. And 9 times out of 10, 100% of the time, the first question I ask a beekeeper, and this will go for every single one of you that is in any program that I'm running, every single time you call me with a problem about your bees, the very first question I'm going to ask you is, what was your mite count? And 9 times out of 10, with new beekeepers, they can't tell me that. And that is a clear indication to me that they are not managing their bees appropriately, because this is paramount. Every 3 to 4 weeks, you have to do sampling in order to understand where your mites are at. Otherwise, you're just rolling the dice at that point if you're not doing this. And you will get into a situation where your only option is a chemical treatment. Now, chemical treatments have 2 different levels. We have soft chemicals that are mostly naturally derived chemicals, and then we have synthetic compounds, and we only have one in the industry, and that's amatraz. So this is how we approach this problem, is through a multifaceted approach to management. It does not just start and end with a chemical treatment. And that's really important part about being a good beekeeper. These chemical treatments, no matter how natural they are, they all have negative effects on your honeybees. And if used improperly, can kill a colony. So they're not something that you should just take lightly. They're something you need to take very seriously, and they have the potential to harm you as well. So not only do you need to take the seriousness for your bees, you also need to take that seriousness for you as well. All right, so that's a little bit about varroa mites. It's going to be the biggest thing you deal with as a beekeeper, at least right now. All right, the next one is another mite. These are tracheal mites, the Carpus woodi. So this arrived in the 80s as well, but this is an internal mite. So this mite actually lives in the trachea of the bees and feeds on the hemolymph, again, the bee blood. It's microscopic. You're never going to see this. So there's no sampling regime you're going to do or monitoring regime you're going to do. It typically affects overwintering bees, and that's because of the longer lifespan of those bees. So your winter bees are going to live a lot longer than your summer bees, sometimes up to 4 to 6 months, depending on where you're at. And that allows these mites to reproduce in the trachea very readily. During the season, these are less of a problem. They definitely exacerbate during those off seasons, during the like downtimes of your bees during the winter. They can cause dead outs. This one of the symptoms you typically see, but most of these symptoms are very unreliable with this particular mite. Is a lot of times you'll see a colony full of honey after they've died out. It's not reliable because there could be other issues like moisture or things like that caused that. So they're very unreliable. Again, it's only identifiable with a microscope. The one nice thing about this particular pest is that some of the control measures that we use for varroa mite actually impact this mite as well. As far as the life cycle goes, this is our life cycle. So it enters the bee and a very young bee, so a 1 to 2 day old bee. It enters through the spiracles. If you recall from the biology lecture, the spiracles are located on the back portion of the thorax and the abdomen, and that's where bees actually draw in air into the trachea. So they enter through the spiracle into the trachea. They're going to lay eggs in the trachea, and then again, you have males that are going to mature faster, 11 to 12 days, and the females are going to be 14 to 15 days after them. They mate with one another, and then your females are going to migrate out to the body hairs and start this cycle all over again. Now, like I said earlier, this is a really this cool electron scanning microscope picture here. But the really nice thing about this particular pest is that the control measures that we use for varroa mite do have some spillover on this. So mite resistant genetics, those varroa sensitive hygienic bees, they can actually kind of deal with this as well from a resistance standpoint because of the heavy grooming behaviors that are imparted in bees that are bred for varroa sensitive hygienic behaviors. It's one of the main things we look for is grooming behaviors when we're selecting for VSH bees. The other thing is that some of the control measures that we use, like formic acid, actually work on tracheal mites. So if you're using formic acid as one of your mite control products, as far as a miticide, this will impact your tracheal mites as well. Let's talk about wax moths. So the greater wax moth and the lesser wax moth. Depending on where you are in the United States, one's going to be more prevalent than the other. You might be one of those lucky ones that has both of them as well. So these are moths that primarily, the big problem with these moths is the larval stage. And the larval stage really causes a lot of damage. So they feed on the protein. So pollen is what they're after. And they can destroy a hive very quickly, weak hives in particular. And this is really kind of the big problem for wax moths. If you have a really weak hive, yeah, they can take it over. Strong hives, wax moths are not going to affect. The big problem that we see with wax moths is stored equipment. If you're storing equipment that has pollen in the frames, that is a primary driver for wax moth infestation. It's most prominent in warm weather conditions. So if you live in warm climates, this is going to be a bigger problem than if you live in colder climates. If you're dealing, especially like when I lived in Southern California, I'm from the Central Coast, it's like 65, 75 degrees year round. That's warm weather. And when you store this type of equipment, a lot of times this can become a really big problem. You see here the adult stage and the larval stages, kind of what they look like. This is the damage that you can expect on your stored frames if you leave pollen in them, and they will just foul these up. Not only do you have all the frass, which is the poop, that's included in all this, you have this webbing that goes underneath all of these cells. So you can see the webbing coming out here. They create these tunnels that are at the bottom of the cells all through your wax, and it will foul up an entire frame very, very quickly. Now, the other thing that it will foul up is your equipment. So not only will it foul up your frames, or your foundation and your wax, it will foul up the equipment itself. So this is the worst wax moth infestation I have ever seen in my nearly 20 years of doing this. And this was a hive that yours truly bought from a commercial beekeeper. We had bought out a commercial beekeeper out of their AZ hives or Aja hives, which are Slovenian beehives that we also work with. And we bought a bunch of new equipment from him. It was new in the box that he had just been sitting on for about 10 years. We bought all that, and he just happened to say, Adam, I've got 3 more hives that had bees in them last year. Do you want those too? And I thought, why not? I'm just going to take them. And that is against my better judgment. And, it's like one of those do what I say, not what I do situations. I always, and for a decade straight would never bring in someone else's bees into my apiary because of biosecurity. We run a really tight ship here, and I let my guard down. And this is what I got. This was an empty hive. But the empty hive, it had died out during the season, and this guy just left it all season long without any management. And this is what you see. So what you see here are cocoons all on. This is the top level of a 3-level Aja hive. So this is a hive that is like a cabinet, basically. All of this white you can see here is all wax moth larvae. And when I pull it away, or pupa, pre-pupa. When I pull this mat away, this is what you see. Now, what I want you to look at is the face of that wall right there. You can see there's all these indentations. Wax moths actually will eat away at the wood as well. What you see below this is a mess of frass. And when I got to the bottom of the box, that's what you see on the right side is all this larva moving around, and that is all poop right there. This took hours to clean and a good scorching with a blowtorch as well before I actually got this thing cleaned up to where it could be used again. But this can cause a lot of damage when it comes to storage. This is mainly a storage pest. So the big thing to keep in mind when you're trying to keep this under control or you're trying to prevent this from getting into your apiary is that you only want to maintain strong hives. Dinky little, hives that you're trying to limp through the season. This is why I tell folks you need to have 2 or 3 hives to get started, is because you don't want just one hive. Because if it turns into a dinky hive that's struggling, these types of pests will take over and you'll be out your hive completely. If you have 2 or 3 hives, you could combine that weak hive with a strong hive, or you could take resources from the strong hives to boost up the small hive, like brood frames and things like that. There's ways to kind of make sure that your colonies don't stay dinky and actually are strong and healthy by having multiple hives. They can support one another. So that's one of the main things, is make sure you have a strong, healthy hive. The other big thing, because this is a storage pest primarily, and if you're dealing with healthy hives, it's not going to be an issue. Because it's a storage pest, you want to make sure that when you store your frames in the winter or in your off season, they don't have pollen in them. Pollen is what they're after. If you store just wax with no pollen in them, this will not be a problem for you. But if you store frames that have wax in them, or pollen in them, you either need to get rid of them as far as culling them or scraping them or something, or put them in a freezer if you're a small time beekeeper and have the space to do that. But you don't want to keep them out during the winter storage period, because that's where wax moths will come in, and that's what they're after, is protein. Additionally, if you expose your stored equipment to light, they won't go after that. So smaller scale beekeepers, I've seen hang their frames in rafters in their garages so that they're open and there's light exposed to them. They won't deal with those things if there's light exposed to them. Again, you can store them in a freezer if you're small time and you've only got a few frames that maybe have pollen that you want to keep for the next season. That's fine. For control measures for basically storing supplies as far as your equipment, paramoth is the compound that we have available to us. It's basically mothballs for wax moth. And you can use that in your stored equipment as well. And typically, the way you implement that, it's a crystallized substance. You put it on like a sheet of newspaper or something on the top of a stack of colonies, and it just permeates through the colony. You do not want to use paramoth in a closed environment, because if you put it in your garage and you store your equipment in there, it is going to smell intensely for a very long time as that gas is off. So something to keep in mind. But that is the compound you have available to you if you feel like you need it. Maybe you live in an area where wax moths is just crazy, and you need something that's a chemical to be able to deal with this. That's your option. Small hive beetles, Athena tumida. This is a problem that is all over the United States, but primarily affects folks in the southern states more than northern states. Now, when I first moved to Michigan 13 years ago, we did not have small hive beetle in the state. We do now have small hive beetle in the southern part of the state, and that's because of the importation of bees from southern states. I am a big advocate of locally produced nukes as a way to secure our industry and make sure that we are not passing pests and pathogens all around the United States. But the way that our industry is built, packages and nukes are moved all around the United States, and with them come a lot of issues, and Athena tumida being one of them. It is a very visible pest. It is a large beetle, and you can see on this picture to the right all of these black dots that kind of look like, I don't know, rat droppings. Those are all of those are beetles moving around those frames. They are black in color, black to brown kind of in color. They do leave a slime trail, and that's one of the main things about them, is as they're feeding in the larval stage, they leave this slimy trail behind them that actually deposits yeast and will ferment your honey and your pollen. So this can foul up an entire colony to where you can't even harvest out of it. And it contaminates that honey with fecal matter. So it's not something that you're going to be able to fix, so to speak, after it's contaminated. They can very, very easily overwhelm a weak colony and can, even in extreme situations, take over strong colonies. They also do have been shown through research to vector things like American foul brood, deformed wing virus, and sac brood virus as well. So it can be a vector as well. This is a tough problem for a lot of folks in southern states, and is a major issue for those folks. And I mean, this is something that a lot of folks in southern states will lose bees to. Where I'm at here in the Upper Peninsula of Michigan, fingers crossed, we don't have this particular pest. Mainly, we believe mainly it's because of the length of our winters. I mean, I'm usually a full 4 to 6 months of winter, and these organisms rely on being able to make it through those cold months to be able to kind of continue to proliferate. And so we have not, fingers crossed, not had it up here yet. This is some more pictures. So this, again, on the right are the adult stage, as you see kind of all around these frames. On the left, this is a formic acid patty, and you can see that in extreme situations. So the thing about Athena tumida is they tend to be, when they're in small numbers, they tend to find like places to hide. The bees will actually kind of regulate them a little bit when it comes to their movements. So when you see them like all over the frames, like you see on the right, this is actually a dead out that I was able to take pictures of that had a huge small hive beetle infestation. I think this was from Virginia. And the one on the left was from another situation I was in, and I think this was in southern Michigan, where I was working with a beekeeper. We were out at his colonies. He had some problems, and so he wanted me to come out and take a look at him. He just so happened to have just finished up a mite treatment, and when I took that formic acid patty off, that is all larvae of small hive beetle underneath that. So they found a hiding place underneath the mite patty, and actually a bunch of juveniles were in there. So they are very prolific, and they do mess up a lot of things. The best control methods that we have for small hive beetle right now is trapping. So things like the beetle be gone sheets is a prime example. There's also oil traps that you can get for this as well. There is a registered pesticide for them as well. It's Guard Star, which is a permethrin. It's a soil drench, so you actually apply it to the soil. And the reason for that is because the life cycle of this particular organism is that in the larval stage, and in the adult stage, it's in your colony. But in the pupal stage, it actually pupates in the ground. And generally what happens is that larva kind of crawls right out that front entrance, drops down in front of the soil in front of your colony, and then burrows down, turns into an adult, and then goes right back into these colonies. So one of the ways that you can prevent this pest from continuing to be able to complete its life cycle is by cutting off that stage of being able to get in, being able to actually go through its metamorphosis phase in the soil from the pupa to the adult. So that's why we use a soil drench. Now, this is highly toxic to bees, but I want you to understand that if you were to use this, you would be applying this in the evening or in the early part of the morning. Once this is dry, it is totally harmless to bees, but in the liquid form, it is highly toxic. Now, another avenue that's non-chemical, that is a barrier method, this would be more of a mechanical control method, would be a ground cover. So you can use things like weed exclusion cloth you can put underneath your apiary, and what that does is it prevents those beetle larvae from being able to pupate in the soil. They can't get through the actual barrier of the soil, of the weed exclusion cloth. I've seen people put down things like carpet, anything, cardboard, anything that you can get to create a barrier between the soil and that organism is a great way to be able to prevent that from being able to pupate in the soil. Another avenue, and this is an avenue that I really like, is using predatory nematodes. So again, a natural control measure. We would consider this a biological control measure because we're using a biological organism to basically impact the small hive beetle. Heterohabdus indica is the nematode that we're talking about here. This is available through a company called Arbico Organics. And Arbico is a company that develops a lot of natural enemies or biological controls that attack other harmful pests in agricultural crops and livestock crops and things like that. So the way that this works, because it's a nematode, and for those of you that are not familiar with nematodes, these are little tiny worms that live in the soil. Some are bad. So some will attack crops, like root knot nematode will attack crops and actually create issues with roots. or they can be predatory as well. And so you can have good ones or bad ones. This one is a predatory nematode, and what it does is it goes around in the soil. You apply it in a liquid solution. So you basically buy the nematodes in a liquid solution, put it in a watering can, fill it up with some water, and then just water the ground in front of your hives. And what happens is that nematode gets into the soil and it starts attacking any kind of insect it can find that's living in the soil. It goes in to the nematode, into the insect, and then reproduces inside of it. So like one nematode comes in and like thousands come out. And so it's a very fast, exponential rate of reproduction for these nematodes. And so over the course of time, you can get excellent control in your soil with this particular product. Now, this does not prevent new beetles from maybe coming in from your neighbor's yard to infect you, but it will prevent the stuff that's in your yard in the ground in front of your colony. So another product that's available to you. The last pest, major pest that I'm going to talk about, and then we're going to talk about a few just minor pests, is AAA laps. Now, AAA laps clary is not in the United States currently, but this is the biggest next threat to our global industry. And the reason this is such a big threat is this is like varroa mites on steroids. It is 10 times worse than any varroa mite you're ever going to deal with. We do not want this in the United States. But the reality is because of global trade, there is a very strong likelihood that this will end up in the US. Probably sooner than later. I hate to say it. So the reason that it's a threat to us in the United States, even though right now it's in Asia primarily, is because Canada imports bees from that area of the world. So they import the Canadian beekeepers have the ability to import bees from Italy, Chile, Australia, and New Zealand. And Australia and New Zealand are in the prime area of potential infestation from this particular mite. So the worry is that a beekeeper in Canada imports bees from New Zealand or Australia, and then they make it across the border, which, is not very difficult because the mites and the bees don't care about any artificial borders. Now, here in the US, we also import from New Zealand, but New Zealand, it's a little bit further than Australia when it comes to the kind of range that we're seeing AAA laps in, but we import from Canada. So, if Canada gets it, we're going to get it. Now, the way that beekeepers in Asia are currently dealing with this mite, when I talked about varroa mite, we talked about we only treat if we absolutely have to. With AAA laps, the current management protocol is that they treat every month. You just treat every single month because of how quick this reproduces and how, I'll say, voracious it is in its abilities. 10 times worse than varroa. We do not want this in the US, but the reality is, again, this is the next thing on the dock, literally and figuratively. So be aware. This is why it's important that you stay up to speed. Every single year you go to a beekeeping conference, I would highly encourage you to go to a pest and pathogen lecture that has someone that's an expert that's giving it, because they're going to keep you up to date on this stuff. This is a really real threat. All right. Last pests we're going to talk about are, let's first talk about vertebrates. Well, no, let's talk about the invertebrates first. So the other insects, ants and wasps. Ants are very likely going to be an issue for you. There's lots of ways to deal with ants. One of the easiest ways to deal with ants is selection of your site. If you put your bees in an area that has a high ant infestation, you're going to get ants. They're going to come after the beehives, not only because there's sweet stuff in there, but there's great places to hide as well and create a colony, like between your top cover and your inner cover. So ants are a real thing. You can deal with ants in many different ways. Diatomaceous earth around the foot or the feet of your hive stand is one way. Tanglefoot, which is a product that we use in agriculture for things like lepidopteran pests, so caterpillars that climb up trees. You put tanglefoot, you basically slap it around on a tree trunk, and they get stuck as they're climbing up the tree trunk. You can do the same thing with the feet of your hive stand as well as coated in tanglefoot. They'll never get into a colony. So ants are very easy to deal with. Wasps can be real tough. So wasps are one of the major robbers that will impact your beehives. So weak colonies that are not able to defend a colony can get overrun by wasps. Depending on where you're at, they can vary from things like yellow jacket all the way up to bald-faced hornets. And that's what you see on this top picture up here. My worst day in beekeeping ever in the nearly 20 years I've been doing this. I was working for Michigan State University at the time, and this is a picture from an apiary that we had in the Upper Peninsula of Michigan, where bald-faced hornets are in very high populations. But this colony was entirely overrun by bald-faced hornets. So all of what you see here are bald-faced hornets, and I was the fortunate one to be at this apiary managing this at that time, and so I got to basically deconstruct two colonies that were completely full of bald-faced hornets. And let me tell you, the amount of stings I got were not fun. It felt like I'd been in a bar fight after I had got done with that, and for several days after, from all of the stings. You're talking about an insect that has a stinger that's about a quarter of an inch long. So my pants, my veil did very little to prevent that. So anyways, but I digress. So wasps are another thing that you can deal with. Keeping a strong, healthy colony is how you prevent that. When you get into the fall, when you get into robbing season, closing down your entrance so that you have a reduced defensive area for the bees to defend against these type of robbers is another way. But bald-faced hornets are actually predators of honeybees, so they actually feed on honeybees. They will actually eat the honeybees themselves. So a very tough predator to deal with. Now, the other ones that you're going to deal with are these that are vertebrates. So like skunks, bears, and raccoons. These can cause major damage in an apiary. And bears, you can see this picture down here. This is an apiary that a customer of mine had that got attacked by a bear, and they make very short work of an apiary. A bear will destroy an entire apiary in an evening. So something you really have to be aware of. And you need to have, if you are in bear country, you need to have an electrical fence. Even if you haven't seen a bear. I had a client probably 10 years ago who we sold bees to, and my very first question, because I knew where she lived, was, when's the last time you saw a bear here? And she told me, oh, I've lived here for 30 years. I've never seen a bear here. Sure enough, the second day after we delivered that colony, it was attacked by a bear because it didn't have an electrical fence around it. If you live in bear country, you need an electrical fence. Bears can smell for a very long distance and will find beehives very readily, especially in the fall when food starts to get more scarce. So make sure you're ready for that. One of the things you want to do is to ratchet strap your hives to prevent, things from getting in. A ratchet strap's not going to prevent a bear, but it can help with skunks and raccoons. So skunks and raccoons are also predators of honeybees. And typically what you see in that situation is that they'll come to the front of a colony and scratch at it to get the bees to come out, and then they just sit there and just eat bees. So if you see scratch marks on the front of your hive or like in front of the hive, like down, like by your bottom board, in front of your bottom board on the ground, that can be an indication that you're dealing with one of those two pests. But, if you have a ratchet strap, you're not going to get one of these to tip over a hive. They're just going to sit out there and eat bees. Some of the things that you can do to prevent them from getting in front of your hives, nail strips in front of your hives. If you are dealing with things like raccoons and skunks, put some nail strips down in front of the hive so that when they do start, get up there, they hit their hands with nails, and that'll keep them from doing that. Mice is another big thing you're going to deal with. Small hives can oftentimes get mice in them. The way that you do this is by keeping the entrance reduced, using mouse guards, things like that. Keeping your hives off the ground on hive stands is another method for that as well. Mice are something that you're going to deal with, especially if you're dealing with like a place that you live that's has real winter. They're always looking for warm places to overwinter as well, and a beehive is a very warm place to overwinter with food source. So keep that in mind. All right, let's talk about pathogens of honeybees. All right, so the first thing we're going to talk about is viruses. So just like humans, there's no treatment for viruses, and Varroa is the major vector for our viruses. Now, we know of over 20 different viruses, and that keeps climbing all the time, that are vectored by Varroa mite after feeding on the bees. And we talked about deformed wing virus earlier. That's what you see on the top. I also mentioned K-wing virus, which is the virus that you see at the bottom. K-wing virus is a virus that affects the development of the hamuli, which is the area of the wings where they connect to one another. And so basically what happens is the bees cannot connect their wings together and they can't create lift. So that's one of the symptoms of K-wing virus. Paralysis is another one that's very common, many different types of paralysis that affect honeybees. Bees that are affected by paralysis tend to tremble, so they actually kind of shake, and you will see that visibly. They look kind of shiny or greasy because they don't have any hair. A lot of times you'll see them crawling on the ground out in front of a colony. They're unable to fly as well. This is an example of a bee suffering from paralysis right here. Oftentimes, you'll see their wings also affected by viral transmission as well, and so sometimes they're like pencil thin. But you can see, you know, we have a healthy bee, honeybee up here above, and then this is a bee that's suffering from paralysis. So you can see it physically looks very different. So if you see something like this in your colony, a lot of times when you actually see this as a symptom, many times it's an indication of very high virolomite levels because, again, virolomite is the one that is allowing this to be vectored within the colony. Another virus that we deal with is sac brood. Sac brood virus is basically a virus that affects development, and that's something you're going to see with all of these viruses. It's primarily affecting development of the honeybees, and so this one in particular, it affects the larva during pupation, and so basically the bee fails to pupate. Now, what you see in the picture here is kind of, I'd say, after this has happened. So basically the way that this works is that after the brood is capped, this is going to die very shortly after, and then after the pupa dies, it kind of turns into this like leathery pupa that is kind of filled with fluid, but it's very firm, and so you can actually very easily take these out of a cell with a pair of tweezers. Now, in this picture, what you can see is that this cell has actually been chewed open. And so what's happening here is that the nurse bees, because this is a dead larva at this point, or dead pre-pupa, we're going to remove this as part of the cleansing behavior of the bees. However, this also affects the transmission of this particular virus. So what happens is that this eventually dries out to this kind of state that you see here, where it becomes kind of like a black or brown color. And when it does get to that stage, it actually becomes quite brittle on the outside. And it's actually loose in the cell. So this will actually like move around in the cell, kind of like, you know, because it's not stuck to anything. But when it hits this stage, which is the scale stage, those scales actually transmit the virus as well. So when these nurse bees are cleaning this out, they're actually getting in contact with the virus and then continuing to transmit that as well. So transmission is a really big thing with this particular virus, and it happens very quickly because if this really takes hold, you know, every time they're cleaning this out, they're transmitting the virus as well. Healthy colonies can clear this particular virus, but if other stressors like virolomite are in the mix, this can have a cascading effect. So it actually can really cause a lot of issues if there's other issues that the bees are dealing with. Hygienic behaviors can help with this a little bit. So that's something to think about. It tends to be that if you have bees that have the hygienic behaviors, that this doesn't get in as high a levels as it does with non-hygienic bees. One of the things that you want to make sure that you understand is the length of time that this virus can last. So typically, this virus will only last for about 4 weeks in materials. So this isn't one of these types of viruses where if you get it, you have to burn everything or get rid of all of your equipment. What you need to do, though, is that if you do have a colony that has been affected by this, you want to take all those infected combs out, and you want to make sure that you store them for a couple months, and that'll be a long enough time for this virus to die, and then you can reuse that equipment. But you don't want to continue to use the same equipment. You gotta break this cycle of this particular virus in order to get through it. But again, like I said, healthy colonies is typically not really a big issue. There are no chemical treatments for this, so the only way that we can get rid of it is through hygienic practices, by making sure that we're clearing out frames that are diseased and have this present, making sure that, you know, we're getting this stuff out of there when we find it. Part of your inspection is looking for, brood that doesn't look healthy. So if you see something like this, you need to start removing those combs immediately and replacing them with fresh combs so that you can get this out. All right, so that was our viruses. A little short section on that. Again, I encourage you to do lots of reading on all of this stuff because there's a whole lot more information than I can put in a hour and a half or 2 hour lecture. The next one is American foul brood, and this particular bacteria. So now we're into the bacterias. This bacteria is why we have apiary inspection laws in the United States. Now, not all states continued with their apiary inspection laws, but many states during the height of American foul brood, which was kind of 1950s through kind of the 1980s, the height of that period, we thought American foul brood was going to collapse the entire industry because it is that bad. It is a bacteria that when you get it, this can be devastating. This is the worst, the absolute most serious thing that we have when it comes to pathogens that we have in beekeeping is American foul brood. The cycle of transmission for this particular bacteria, the larvae are fed spores of American foul brood by nurse bees, and the spores basically kill that organism as it's developing. Now, the thing about this particular bacteria, and we're going to talk about another one here in just a moment, European foul brood, is that this particular pathogen kills the larva after the brood has been capped. So as it starts to move into that prepupal stage, that's when it kills this. The difference with European foul brood is European foul brood kills the larva before it's capped. So that is a key symptom difference between these two pathogens, between these two bacteria. Now, when it comes to American foul brood, and this goes, true for European foul brood, is that a lot of times it's the movement, it's the transmission of this that creates the big problem. So with American foul brood, it's the house bees that are trying to remove these contaminated larvae, trying to clear out these dead organisms that really start to move this around. It also moves through the colony through food stores as well. So it's not only through contact between the bees and these contaminated larvae that are dying, but it's also through the food as well. Things like robbing also causes spread. So if your bees are in an apiary with a colony that has American foul brood, anything that comes into that colony and comes into contact with those spores is going to bring it back to their colony. So this can take over an apiary that you're not paying attention to very, very quickly. Drifting, your bees drifting to another colony can also cause this. Now, the big problem with American foul brood is not only the virulence of the actual pathogen, but these spores, these American foul brood spores, can live in woodenware for up to 80 years. This is why we don't recommend buying used equipment, because you have no idea, say Johnny inherited grandpa's beekeeping equipment that was been in the barn for 30 years and nobody remembers why grandpa quit beekeeping, but turns out that grandpa got hit with American foul brood in the 80s and lost everything and then just threw it all in the barn. You were to pick up that equipment and start using it, American foul brood is going to be right there with you. This is why buying used equipment is very, very sketchy, especially for new beekeepers who don't know what to look for and what to ask for. And even as experienced beekeepers, a lot of times we're rolling the dice if we're buying used equipment. So don't buy used equipment. That's the best way to prevent yourself from getting American foul brood. Now, I did say that this is the most dangerous and serious and contagious disease that we have in honeybees, and it can kill an entire operation, but I don't want you to feel like, oh my God, what am I going to do? Because American foul brood is not as prevalent as it used to be. So in my nearly 20 years of beekeeping, I have seen American foul brood, real American foul brood, not just stuff people call American foul brood, because you got to test in order to be able to determine if it's a real AFB. Twice I have seen that in my entire career. Once in Michigan and once in California. It's the only times I've ever seen it. And so I see a lot of beehives, because not only do I work mine, we have customers that I work their colonies for. We run programs through universities and colleges and nonprofit organizations, and I see a lot of bee colonies. So the likelihood is this is not something that you're going to be incurring, but it's something you want to be aware of. Okay. The symptoms of AFB. Sunken cappings are a prime symptom. The cappings sink when the bees, when those organisms die within the cell. Perforated caps. This is a situation you see here. So you have sunken caps here, and then you have perforated caps here, or caps. This is actually they're uncapping right now, but a lot of times you'll have holes in the caps that are just perforated there. You see that these up here, they're trying to uncap these, and they're going to start removing this diseased, these diseased larvae out of here at some point. But again, that passes the disease. Spotty brood patterns is one of the things that we see because you end up getting a lot of these dead cells, and so this would be considered spotty because you've got a lot of dead cells that are in there. One of the main characteristics for American foulbrood is the smell, and it has kind of a sulfury smell to it. If you've incurred it anytime in your career, it's not a smell you forget. It's very different from the smell of a European foulbrood. It has a very distinct odor. This is a very bad pathogen, though. So light brown in color is kind of what you see when the actual goop comes out. So I'll show you that here in just a slide or two, but just to reinforce that this is a healthy brood frame on the right. There's your AFB on the left. So it looks very different. The way that we test this, if you feel like this is something that you're incurring, well, number one, I encourage you to talk to your State Department of Agriculture. All state departments of ag want to know if AFB is in their state. Because again, when you incur this, this is the most dangerous and contagious disease that we have in the industry. And state apiarists have done a really good job over the last few decades in getting us to a point where this is not to a point where it's going to collapse our industry. And we have AFB very under control at this point. Again, you're likely not going to incur this, but you need to know about it. But if you do incur this and you need to do a test of this, the way that we test for this is called the matchstick test. And what you do is you basically take a matchstick or something like a matchstick, poke it into the cell that you believe is contaminated with AFB, give it a nice stir, and then slowly pull it out. And if you get this ropiness that you see here, where this is distending and you see the brown kind of coffee color to it, that is a key indicator of American foulbrood. That's the easiest way to test for it out in the field. And there are other tests available for this, but this is the easiest way to do it that doesn't require you buying anything. So that's one thing. But like I said, again, you're likely not going to interact with this. What is the treatment option for AFB? There's only one option, and that is to burn everything. You do not want to get this. You don't want to waste your money on used equipment and find out you got AFB and have to burn it all to the ground. That is the solution for AFB and the only solution for it. Not something you want in your apiary. Preventative measures. Never buy used equipment. And this tends to be a hobbyist issue because you're starting in a new field and you're just looking to save a buck because it's expensive to get into beekeeping. I get it. But it'd be terrible if you bought a bunch of equipment and had to destroy all of it. Never let equipment that's not yours in your bee yard. So this comes down to biosecurity. Don't let other people's hive tools in your bee yard. Sanitize your hive tools when you interact with diseased colonies, whether it's American foulbrood or anything else. 10% bleach solution and soak those hive tools. Frames. Don't let other people bring frames into your operation. If you are buying nukes from someone, make sure you're buying them from a reputable source who's not selling you garbage that's diseased. Most nuke producers that are good at what they do, that have been doing this for a while, would never let something like this come out of their apiary. But new nuke producers who don't know any better and maybe only have a couple of years of beekeeping under their belt can realistically, this can transfer into your colony, into your apiary from buying bees from somebody. Again, if you suspect it, use the matchstick test and react immediately. This is not something to wait on. If you identify that you actually have AFB, you need to burn that colony today, right now. Don't wait, because the longer you wait, the more likely you are going to have that colony passing that to other colonies or robbers coming in there, because it is a dwindling colony when it's got AFB. It's a sick colony. And so robbers are going to come and take advantage of that. And they will pass this on to those colonies that they're from as well. This can take out an entire apiary in a very short period of time if you do not deal with it. All right, so the next bacteria that we're going to talk about is European foulbrood. So European foulbrood Melissococcus platonis is another bacterial disease. Most of the time when we see this, it's a lot of the time it's in the early part of the year. It has a lot to do, we think that a lot of susceptibility with European foul brood has to do with stress. So bees get under stress and they're more susceptible to this. And that's one of the stages that we typically see in the early part of the year, but it can persist through the entire year if you don't do anything with it. The bees, the larvae die in the larval stage, in the uncapped stage. And one of the easiest ways that you can tell, because this is passed through the food, one of the easiest ways that you can tell that a larva has been contaminated is by the color. So you see all these different, you see like this one right here, that little larva right there. That is bright white and very good looking as far as the food is concerned. You see the one right next to it, that yellow color. The yellowing is a prime symptom of contaminated food by EFB. And you see that that will also translate into later stages of the EFB where it advances, where you actually see that discoloration in those dead larvae that are twisted to the bottom of the cell. To kind of reiterate the stages of EFB, so again, the symptoms are going to be, you're going to find these uncapped cells. You're going to see twisted larvae in the late stages of EFB, and they can appear dried. When you do see these, if you were to do a matchstick test on these, you would get no roping. But they do also have a sour odor. It's not sulfury. It's more of like I kind of think of it like a cheesy kind of rotten cheesy smell than I do the sulfur smell with AFB. But you can see that this is kind of the stages of EFB pictured here. Starting at the top left, you see that bright white, nice, healthy larva, and then it starts to discolor over time, and it's starting to move around and twist a little bit. By the time that bacteria really takes hold of the digestive system and really just disintegrates everything in there, you get to this stage where it starts to slump down to the bottom of the cell and really become twisty and will eventually become kind of scaly when it comes to this end stages of that. So if you're seeing something like this, again, open cells, like big difference, capped and open between AFB and EFB. If you're seeing this in open cells, this is EFB. That twistiness, you don't see that in AFB. The twistiness is very indicative of EFB. Control methods for EFB. Sanitation is probably your best bet. Culling old brood frames, making sure that if you are interacting with contaminated hives that you're disinfecting those tools. Terramycin is an antibiotic and is one of the things that is available for you for EFB, but it is a preventative measure. It does not cure infected bees. So if you have a colony that has EFB, this will not help you. It will only help with preventing new infection in a colony. It does require a prescription from a veterinarian, though, and that's part of the veterinarian feed directive that we have here in the United States. Now, the ways that we deal with this, as far as a control measure, there's really only a couple. And I mean, it's very similar to EFB, but not as bad because the spores don't persist like EFB does. So one of the things for the length of time that EFB does, but one of the things we do have to get those spores out of the colony. And so one of the one of the measures that we do if we interact with a colony that has European foul brood is what we call the shook swarm technique, which is essentially where you take all your bees and you shake them out onto brand new equipment and get that old equipment out of there. One of the measures that you can use for this, and this is actually from the UK, this particular particular recommendation, and I really like the recommendation, is why I'm recommending it to you, is that if you have less than 25% of infection on 6 or more frames, meaning that when you kind of look at the overall picture and you have 6 or more frames of bees, if you have less than 25%, do the shook swarm, meaning that, get those bees off of that equipment and get that equipment out of there. If you have more than 25% infection on a small colony, you should destroy the colony. This will persist. This will move from colony to colony, and you need to get it under control very quickly. If you catch this very early on, just removing some frames very early on sometimes can get you out of this. But that shook swarm technique is the technique that is the recommended technique for really getting out of this. And if you're dealing with a smaller colony, you're going to catch this typically early on. Large, healthy colonies don't typically get affected by EFB. It tends to be colonies that are under stress, at least from my experience. But strong colonies can be susceptible to this if they're exposed to it. But that's one of the things to think about is that, if you do have a small colony, is it worth risking all the rest of your colonies to keep that dinky colony around and try and limp it along? Now, get that colony off of that equipment, do a shook swarm, and start fresh without the EFB. All right, now we're going to deal with fungi. So we're out of the bacterial diseases at this point. So fungi, we're going to talk about Nosema apis and Nosema cerana. So Nosema apis and Nosema cerana are two microsporidian parasites. These are fungus-forming spores, so microsporidian. The spores are spread through contaminated food and water, and they germinate and reproduce in the midgut. So this causes a lot of infection in the gut as well. Now, the symptomology that we typically associate with Nosema is diarrhea, so dysentery, but that is only true for Nosema apis. Now, the thing that you need to think about when it comes to, and we've talked about this with a lot of the other pests and pathogens, is this ability for these things to reproduce at rapid rates. A spore that gets into a bee in about 2 weeks will create about 30 million spores. So this really, really takes off very quickly if a colony is being infected by it. All right, so let's look at the symptoms. So symptoms of Nosema apis. Now, I will tell you that Nosema apis is not the primary disease that we have, the pathogen that we have anymore in the United States. It used to be that Nosema apis was the most common. Now it's Nosema cerana. Now with Nosema apis, a lot of times you would see that bees would just stop eating. So they wouldn't even take sugar syrup. But one of the main symptoms that you see is this picture here. And this is just pure dysentery, fecal matter all over the place. Bees will not defecate in a colony generally. So over winter is typically when you're going to see this kind of concentration of this. You'll also see the poop all over the front of the colony as well, all over the front entrance of the colony. But one of so that's one symptom is you see all this poop everywhere. The other one is that you oftentimes will see bees that are shaking or dragging legs or just stumbling. They can't fly. This tends to be something that Nosema apis tends to be something that affects bees in the cooler months. So it's typically something that we see popping up at the late part of the season through the winter. That's Nosema apis. Nosema cerana, you don't get the dysentery and you don't get the crawling. This is more of a thing that can take you by complete surprise and you may not even know you even have it. Sometimes you'll see dead bees around, but it's not reliable. But basically, the main symptom for this is just kind of an overall decline. And this can happen any time of the year. So, if you got a colony that's declining and you know it's not Varroa because you've done checks and you know that your Varroa is in check, you see that nothing else is in play, it might be Nosema. Now, Nosema is not something that you can generally diagnose on your own. It's something that has to be diagnosed with a microscope. So that's something to keep in mind. But it's something you need to know about. Now, the impact of Nosema overall. Nosema reduces the lifespan of bees by about 50%, can reduce your honey yield by almost 50%. It reduces populations of bees because it impacts the salivary glands function in nurse bees. So it actually impacts their ability to feed other bees. It ages bees faster because of hormonal development disruption. It can cause bees to actually become foragers earlier in their life cycle as well. And it just completely disrupts the enzyme production in the gut. And actually, the result is starvation, is basically what you see in your bees. So this is really a silent killer. Best practices for prevention of Nosema: maintain healthy colonies. And that's one of the things we're going to say for everything is just maintain healthy colonies. It's why you do inspections so regularly. Rotate your combs. You always want to be rotating your combs out. You should always be culling combs. And I generally cull combs every 3 to 5 years, or about 20 to 25% of your combs every single year. Good ventilation in winter is something that can help with Nosema prevalence. Reduced exposure to straight line winds, the stress that's associated with that. Good sun exposure, another strategy for helping with prevention. Don't move your bees around too much during the winter. This causes stress, and that's what we're trying to reduce, is the amount of stress, which can make them susceptible to this. And you have to replace soiled combs. If you've got combs that look like that picture I showed you earlier, you got to get rid of those combs. Don't keep stuff that's been pooped all over because it is going to pass that pathogen right on to the next colony you use those frames for. Control methods, as far as the chemical control methods, fumagillin B is what we have. It's an antibiotic. It's fed as syrup, and really wide reports say that it can be. It really is kind of ineffective. I'll be real honest. It is what this product is. One of the things this product is labeled for, but there's a lot of reports that it's not really that effective. So again, something that you want to keep in mind. The apiary, the veterinary feed directives do require a prescription for this as well, because it is an antibiotic. Follow best control practices and preventative measures is really your best bet for control of nosema. Chalk brood. So chalk brood is a fungus as well. Chalk brood is what you see on this right picture here. You'll see these kind of mummies right here, and they'll be either white or black. They're typically found on the bottom board of the hive, and a lot of times you might even see them out in front of the hive if bees have dragged them out. Now, this won't take down a strong colony, but it has an additive effect. And so if you're dealing with weaker colonies or colonies that are compromised, chalk brood can become a big problem. Way do you control this really is keeping the hives dry and in a sunny location. You need to get rid of any combs that may have a lot of this infection or this fungus in it. But there's no registered fungicides to deal with this. This is something that a lot of times it's a removal that really comes into play. So I'm going to leave you today with an emerging threat that is, well, it's really knocking on our doorstep. And so it's something we need to really be aware of as beekeepers here in the US. So the emerging threat that our industry is facing is a new mite called Tropalaelaps. Tropalaelaps is similar to Varroa in some respects and not so similar in a lot of other respects. So it is an ectoparasite, just like Varroa. It's about a third of the size of Varroa, so much smaller. And its natural hosts are Apis dorsata, the giant honeybee, and Apis florea, the dwarf honeybee. Comes from Southeast Asia, and one of the big things that's different about this than Varroa is that it has a shorter life cycle. So instead of 10 days like Varroa, it reproduces in 8 days. So because it has a shorter life cycle, it reproduces at a much faster rate than Varroa. It's much more fast moving than Varroa as well. Varroa is a pretty slow parasite, and that's one of the reasons why bees that have Varroa sensitive hygiene bred into them those behaviors. That one of the behaviors that is looked for as they look for these behaviors in breeding for Varroa sensitive hygiene, one of them is grooming. And so like a slow moving mite can easily be groomed off with a lot of grooming behaviors incorporated into a bee. And so that works really well for Varroa. Unfortunately, this mite is much faster. And so things like that grooming behavior aren't nearly as effective on this mite. The other thing that's really different about this mite compared to Varroa is that it feeds on multiple areas of the immature bee. And what I mean by that is that with Varroa, typically we see one major wound, and it's typically right around the fat body is where we see, the vast majority of activity with Varroa is feeding around the fat body. This mite feeds everywhere. And so it's like instead of having a one spot in your body wounded, multiple areas of your body are wounded during development, which impacts many, many more areas of the bee rather than just the fat body. And so it is a much more devastating parasite to bee development than Varroa. The thing that we're finding in research right now, and this is all brand new stuff. This is another example of why science is so important and why funding science is so important, because this is a new threat. We didn't have this on our radar before because it wasn't on our bees. It wasn't on Apis mellifera yet. And so this underscores the importance of funding science to make sure that things like honeybees we're able to take care of them and keep them. The thing about Tropy is that what we're finding is colonies that are infested with Tropalaelaps usually collapse within 30 to 60 days of infestation. That is a tremendous collapse. And they infest at higher rates than Varroa. Like I said, the life cycle is shorter, and so they reproduce at higher rates. They also reproduce more offspring than Varroa, almost double the amount of mated females that come out of a Varroa infested cell. About double of the tropicalaps will come out. So they're reproducing at a much faster rate. It also has an additive effect with Varroa. So when you have both of them in a hive, the colonies collapse even faster. So this is a really serious issue. This is going to be it's going to be one of those points in history that we look back in apiculture as a turning point in beekeeping. And by a turning point, that might be positive or negative. I'm not really sure at this point. But right now, we are facing a huge uphill battle. The only treatment that is working for tropicalaps that they're finding in the areas where it is currently at is formic acid. Now, if you're not familiar with formic acid, maybe you're new to bees. Formic acid is a organic acid. It's very effective on varroa mites, but just like all miticides, there is a downside to this chemical. You can sterilize drones by using it. You can cause supersedure by using it, especially in warm climates. And so when you think about it, if formic acid is the only chemical that you can use to control this, and you are a beekeeper in California in the middle of the summer, when you're hovering around 95 to 100 degrees all summer, you can't use formic because it will kill your entire colony. It can't be used over 85 degrees is what the label says. And if you go over that, it becomes toxic to bees. So, for example, some areas of the country cannot use formic acid. The warm areas of the US that are warm during the summer when bees are really motoring, you won't even have access to a control measure to control tropicalaps because the chemistry that we have available will kill your bees in extreme heat. So this is a huge issue for our industry. And like I alluded to earlier, varroa sensitive hygiene has really little impact. All of these breeding efforts that we've been doing since the early 90s, all of these breeding efforts that have been going into developing bees that can deal with varroa through varroa sensitive hygiene, a big portion of which grooming is a component of. The grooming behavior doesn't have any impact on tropy. Tropy is much too fast moving and much too small to be handled by grooming behaviors. And so VSH bees, the bees that we use for varroa, for good varroa control, are not even effective for this. So this is a very scary situation that we're in. In my beekeeping career, which spans 20 years, this is the scariest time I have ever seen. Those who were before, when varroa mite first came into the US, that was a really scary time. But this is the scariest time I've ever seen in my 20 years of keeping bees. And this is really serious. So you need to know about this stuff. I don't want to, completely, make you feel like doom and gloom, but I am a firm believer in living in reality. And the reality is that this is a very serious problem. It's currently in Southeast Asia, primarily, so that's its native range. It is also found in Korea right now, Russia, and parts of Eastern Europe. And just as an example, this was found on a container ship in 2026 and to 2025, headed for New Jersey. And what they found was a swarm on a container on a ship. And that swarm had tropicalaps. So thinking like, well, we just won't import bees, it has nothing to do with the spread of invasive species. Invasive species, plants, insects, everything biological that is called invasive, moving into a country that it's not native to, that is all done by global trade. Lots of critters live on container ships and all of these types of things and go across oceans. This is going to hit the US. This is going to hit Canada. This is going to hit the entire North American area at some point. We hope that over time that our scientists have enough time to develop the tools we need to handle this and the strategies to monitor for it. Because right now, monitoring is still an issue with this because the traditional monitoring methods that we use for varroa don't work for tropicalaps. So something to keep your eyes on, but underscores why it's so important to continue your education and continue building that education. This is a lifelong endeavor you're going on. If you're really serious about bees, like this is a commitment. It's not just taking this program and thinking that that's it. There's a lifelong commitment to learning, to being a good beekeeper and keeping your bees alive. What I'm going to leave you with is just some final thoughts. So, beekeeping is a fun and rewarding venture, but you, as a beekeeper, need to take your education seriously because, like I said in the beginning of this, you're in a totally different era of beekeeping than a lot of other beekeepers in our history here in the United States were exposed to. Now, you're going to struggle your first 2 years when it comes to pests and pathogens, but stick to it. Keep doing the work, keep studying, keep exposing yourself to this topic, and just get familiar with these things. Again, there's not that many to understand, and so getting familiar with them is not really that big of a task. You want to document everything when you're going through these colonies as far as your inspections because it's going to be a big data point and big ability for you to make those kind of historical correlations and be able to understand when potentially your bees are dealing with pests or pathogens and more prevalence during the year, which allows you to kind of develop a preventative strategy for them. Don't wait to treat the problem. When you see a problem, you need to act. And the further north you live, the faster you need to react because the seasons are that much shorter. So you have less time to work with this. So take this stuff seriously and take action when needed. And lastly, knowledge is power. Keep studying, keep exposing yourself to this topic, keep up on what's new and upcoming. AAA Labs will likely be here at some point. You want to make sure that you know when it's in the United States when it gets here because it's going to change everything for us. All right, folks. Thanks so much. We'll look forward to seeing you at the next lecture.